We are excited to have you here, and welcome to everyone who's joined us online this afternoon. Great to have you. A little bit of a gray sort of day out there. We're getting used to it, though. This is going to be the new sun for the summer of 2023. We're thrilled to have you with us this evening to introduce you to two of our visiting scientists who are with us this summer at MDIBL. We are so excited to have them, Dr. Rebekka Schneider and Dr. Rafael Kramann, who come to us from Aachen, Germany, and they are leaders in tissue fibrosis. They are really doing some very exciting work to kind of bring all of these sophisticated biological tools together to address some of these very complex problems. And so we're excited to have a conversation with them this evening, learn a little bit more about the work that they're doing. So we want to encourage all of you to ask lots of questions. We want this to be a nice participatory conversation. So with that, I'm going to turn it over to my colleague, Fred Bever, and we look forward to learning more about this amazing work. Thank you so much.
Thank you, Jerry. It sounds like the mic is working. Welcome to everyone in the Marin Auditorium and in the Zoom auditorium. I'm glad to see you all here today. So these distinguished physicians and investigators from Aachen are working with this ensemble of bioinvestigative tools: genomics, proteomics, epigenomics, single cell analysis. There's dozens of them. We don't even know how many, right? It's a mystery. But if you think of it as an ensemble, what are they? What are they about? How do you explain what these tools are?
It's basically just you can study the constituents of the cell, basically. So the protein that is in a cell, and this, when there's like injury to the cell, this protein in the cell changes. So this is proteomics, and you can actually look at signaling. So like certain proteins get phosphorylated if they, there's a signaling pathway activated. This is called phosphoproteomics, when you look at the parts of the proteins that are phosphorylated and where. And then genomics is like a really big thing where you can look at, the DNA in a given cell, but also at the transcriptomes and the mRNA that is being produced and then translated to the protein. And there's really like a revolution now with the single cell technologies, that you can actually not just isolate tissue and look at the, proteome or the transcriptome or the genome of the entire tissue, but you can actually isolate single cells and then have this information from every single cell. And this helps you really to dissect mechanisms of, homeostasis, but also disease, how cells change in response to injury, how they crosstalk to each other, how there's different cell states when cells differentiate towards injured cells, towards disease-driving cells, towards cancer cells. There's all these really things you can do now that really change our understanding of tissue homeostasis, but particularly of disease.
Can you think of it like a multi-track tape recording of an orchestra, where there's a microphone on each instrument, something like Peter and the Wolf were saying, on the oboe and on the clarinet and on the flutes and on the piano and the timpani, and you hear it all at once, but then you can separate them by listening to each different tape track and then put it back together again. Is that a decent way to think about it? genomics, so you're looking at the genome.
Absolutely, yeah.
You're looking at the RNA that's produced from the gene. You're looking at the transcription that then becomes proteins and expression, but much more than that.
No, absolutely. I think this is actually a good example. I think you need everything together, that the cell works, that you have the entire song, or you hear everything. And this is basically the same. You have all these different layers basically that help you to really understand what the cell does in response to certain stimuli.
And I think the important thing is that you want to understand what's kind of then wrong. So something has gone wrong and you can't directly hear it, for example, in your analogy. And this is something you can use these tools. So you don't want to have it's not the, let's say, perfect piece. Something is wrong and you want to figure it out. And I think that's the most important. So you're not looking at homeostasis as something that's absolutely as it's supposed to be, but we're always comparing the state of this is like how it's supposed to be versus there's something wrong. And it's like something tiny, because they're all kind of, there are a lot of experts and so really seeing tiny differences. I think there's a...
Dr. Haller, please.
Advocates. These are so many molecules. You have just told us no numbers, but is it really possible to understand this all? How many proteins are changing, or cells are genes are changing in cells? What amount of data are we talking?
And just to restate this for the folks who are joining by Zoom, Dr. Haller has asked, this is so much information. There's so many things different going on at once. How can you possibly understand that kind of complexity?
No, very good question. It's like I think it's like you never understand everything at once, but you can use all these different, data sets you get to model, to build a computational model that tells you what is happening. And then the key is actually to validate this model, right? So you have to, and you cannot not validate everything, but there's, certain disease driving things you identify where you think, okay, this could be something where you can intervene with the therapy. So this is something where you can build a small molecule, an antibody, whatever, novel therapeutic approach where you can intervene. And then you have to validate this really in the wet lab, using animal models, using organoids, any kind of tools to say, okay, this computational model predicts that this is happening. Certain molecules talk, in signaling pathways from crosstalk from one cell to the other. And then this is something you have to validate. And you just have the computational model. The computational model is imperfect. And also all the tools are imperfect. You never detect everything. So certain tools, particularly when you look at single cells, are very sparse. So you just identify around 3,000 to 4,000 mRNAs in a given cell, but there's 20,000, 30,000. So it's just helping you to build computational models, and integrating all these different omic layers help you to get a better picture. But there's always things lacking in the model.
So you start with a computational model first rather than an animal model. Okay.
Yeah.
Or a cellular model. Sometimes both ways.
Yeah. You can actually use human tissue. You can use any animal model and analyze this with omics. But our idea is like to really be translational. So we want to actually learn something. Even if you do animal models, we want to learn something that at the end helps us to treat patients, right? So and that's why we usually very often start with human, or we could start with an animal model, but then always try to link this to human. So to link this to compare, mouse or other organs or other organisms. And I think we're going to talk about this later, to human disease. And so this is basically you can start either with human or with the animal model.
Dr. Haller, was there a particular point that you were trying to elicit?
Personal level of data. Do you think that you both work on Rebekka works on bone and you work on the, so you think that in the evening and afternoon you said, well, see that, this omics array, does it look like yours, or how does it work?
Yeah, I'm not sure if I should say that's unfortunately the case or that's fortunately the case. But yes, that's exactly how it is. So you have these large data set, and then, we always have our validation in a different organ. So for example, you see something and you think this is really interesting, and this seems to make sense also on a bigger scale in different tissues. It's like a mechanism where you think this would make sense across the organs. And then it's like nice if you basically can turn around and say, how does that look? I mean, I look into the bone marrow, and then you ask, how does it look in the heart or in the kidney? And we are often very surprised that exactly if you look at a mechanism or a gene, they also come up in these data sets. And I think this is always like something that makes us feel we're going in the right direction because it's something that's conserved. It seems to be important. It's not something because, yeah, some artifact or something because the sample was taken in the morning and the other one in the afternoon, or like the mouse was 5 weeks old or 8 weeks old or 24 weeks old. So I think it's important to test your models and also across the organs.
But it's actually, if I just add, it's just not us. We have like a large team, and it's like really computational team. I think in my team are around 60 people. Rebekka says over 30. And there's like really computational experts, bioinformatics, computer scientists that really help us to analyze the data. It's nothing I mean, we do coding.
It's more us than asking someone to take this, but yeah, that's the same.
So for those of us who are lay people, when you want to understand a heart attack from all of these different layers, or when you want to understand kidney fibrosis and other organ fibrosis from these multiple points of view, what are you physically doing? I think I get that you're interrogating data sets that are already existent, but what else is your team doing? Maybe with actual physical models, with the mouse or with the Petri dish. What do you have to actually roll up your sleeves and do in the real world?
Yeah, you actually have to. There's data sets out there, but still not the perfect ones. You have to generate data sets. So, and then usually you look for an animal model of choice, or what we try to do is like having an animal model and actually human samples. So you basically try to talk to other investigators and biobanks to identify human samples that are from a certain disease that really reflect what you want to study, like a heart attack, different time points after heart attack of patients, and then gender balanced like cohort of patients. You get the tissue, and then you can really think about what layers, omic layers, would actually help you to understand diseases. So one example would be to take a cryosection of the tissue and then use this for looking at morphology, looking at spatial expression of genes, but then also isolate nuclei out of this tissue and then subject these nuclei to certain other technologies, like looking at single nuclear transcriptomics, looking at single nuclear genomics, looking at proteomics. There's a lot of things you can do.
And are you working on the same actual sample, or just typical samples that you can put through different processes, but they're still?
So ideally, I mean, there's like all kind of approaches, people, and this is also developing really rapidly. So ideally, you have the same sample where you can do all things at once.
So maybe this is a good time to start looking at some of your slides. But in terms of all of these omics, if to stick with the ensemble or the orchestra for a moment, is there kind of the lead instrument? If let's say you guys, the conductor, who are you calling on the most? Is it just the ability to process all of the data?
Yeah, I think one thing we haven't talked about is once you have these multiomics and you identify this interesting pathway or gene where you think this is important for disease driving, you have to validate it. So basically that's the moment when you have your multiomics, and then you go to the next model, and you can do this either in a cell culture dish, for example, and then we have models in the dish to induce a disease. And then you, for example, you take the gene that you think is interesting and you overexpress it, so you want it to be expressed more, or you want it not to be expressed, and then see what the phenotype is, because that way you figure out if you do this, this could be a drug, basically. Or sometimes you're lucky and there's even a drug somewhere out there that you can add to your cell culture dish. You can use a mouse model, you can use zebrafish, all these different models that would mimic your disease. And then you kind of show how, if you modify it, if it leads to kind of like, let's say, a better disease outcome or even worse disease outcome. And that shows you then that this particular gene really does play a role. And it's not just something that in this pool of data you identify to be interesting, but you really have to narrow it down. So you start with like this bunch of data, and then you go down and you want to narrow it down to this really a function in the end.
Yeah, I think the working horse is probably becoming like single cell transcriptomics, where you have, mRNA of individual cells out of like a tissue that you digest, or where biobanked frozen tissue where you can isolate nuclei. And there's now even some new technologies where you can do formalin fixed paraffin embedded tissue from pathologists, and you can really isolate nuclei and look at transcriptome of any given cell in the tissue.
So you can see what the exact function and structure, genetic structure of each individual cell is.
Yeah, what the cell basically, for what mRNA does the cell actually produce at the time point where the tissue has been fixed, which tells you, and then you have a large cohort of, different time points, and this really tries to, you can then build computational models of certain cells that get in these patients in disease comparing to healthy that get injured, they de-differentiate their signal. And so based on this, then what Rebekka mentioned, we're really trying to identify mechanisms that we can intervene for potential therapies.
And you're creating these actually spatialized maps that show these various activities going on during a heart attack when fibrosis develops. Can we say that you actually are doing this through the fourth dimension as well, through time? So you'll take a sample from 1 second, 1.1 second, 1.2 seconds into an event.
Yeah.
Okay, so maybe you could show us a little bit of what the maps look like. And they have actually created these maps. They've found targets for therapies. We'll ask later about whether any is getting into clinical trial. But maybe you could show us some of these illustrations that you've created. And I would encourage everybody, in addition to Dr. Haller, to jump in at any point when you have a question. But please, Dr. Haller.
What you're describing, I can hear the word. I know a little bit what you're talking about, but to be honest, I don't really know exactly what you're talking about, because when you're like me, trained to look at a patient or perhaps to look through a microscope, it took me forever to find out under the microscope what this information means. But now you're using something which is very different. So you need another training to do this, isn't that the case?
Yeah, exactly. I mean, it's like, there's some that actually code, so you need some understanding of computational technologies and tools, and you need to really try to speak the language of computer scientists a little bit, even as a life scientist, and this really helps. And this was a learning curve, certainly when we started that, because initially the computer scientists, they just talk about numbers, right? 1, 0, 1, 1, 0. And we talk about, proteins and disease mechanisms. And this is really something then when you meet with your computational team, there's like, some biologists or like some physicians on my team have really some computational skills. And there's some really hardcore computer science people, and they all together, actually, this team together, speaks like or lifts this like to a new level. And this is really like something for the last 5-6 years was also like a learning curve for us to understand how you talk to people, how you tackle, how you answer certain questions, what tools, what options are there. And I think 5 years back we couldn't really do this, and there was really tremendous team effort also from us learning things that we didn't know before.
Maybe just if the folks watching on Zoom, are you able to hear Dr. Haller's questions? Because I imagine he'll have some more, and we could bring him up. We have an extra chair. Any answers on that, Anna? Yeah.
I think bringing him up would help.
Vision. Oh, sorry, did you get an answer from Jane? Sorry.
If he speaks right into the microphone, I can hear him.
Do you have a microphone?
No, it's above.
And I don't.
Oh, it's above. Okay. Well, I think you need to speak up too, Jared.
So my question is simply, I think we all can envision why fibrosis in soft tissue is a problem, right? That it impedes function. I've never really thought about fibrosis in bone, Rebekka. Can you give us an example of how that's caused and what happens when, I'm sorry, there's rotation.
No, that's great. I think this is exactly what we will show in the slides, and maybe we just that's a good way to introduce the slides, and then we can answer questions, particular questions later. Yeah.
Okay, so maybe I start. So basically, I mean, my lab actually studies fibrosis in mostly in the kidney and the heart. And what you can see here is actually this. Here's like a kidney, and this is the heart. And you can see this is a trichrome staining. So all the blue stuff here, this is fibrotic scar. So this is scar deposition, and our data suggests that basically this scar deposition destroys this blue stuff here, destroys the architecture of the organ, and then really drives kidney failure. And the heart, this actually stiffens the heart, so the heart can't pump as good anymore. And it also triggers electric instability because it doesn't, conduct as well as other tissue. And then you get sudden cardiac death from arrhythmia. And this is actually a bone marrow. Maybe Rebekka can explain this better.
And for the bone marrow, you do a different staining. It's a reticulin staining. It does basically the same. So, I mean, we have to imagine our bones are not just, so that we can move and walk and raise our hands and so on, but our bones are full with blood forming cells. So that's the most important thing. So, I mean, here it's really about the blood in the end, although we look at the bone. And as you can see in these pictures, all these like tiny dots here, these are the blood forming cells. And then in this particular disease, you see these black fibers, and you also can see new formation of bone. So at some point, the fibers take over the bone marrow, and there are basically no blood forming cells anymore. And that's really the problem. So it's about making blood, not about the bone structure by itself.
Could you maybe talk a little bit about, first of all, just where this fibrotic matrix arises, what leads them to arise in the first place?
Yeah, so it has been very controversial, so like this is like the scar, you can see that in blue, and where are the cells coming from that produce the scar? That has been really controversial. And the cells that produce this matrix are the myofibroblasts, and most of all the data sets now, including ours, suggest there's resident fibroblasts and pericytes that actually produce most of this matrix. And there's not all these other processes that have been discussed from epithelial cells and endothelial cells that they don't exist. So it's basically, I mean, cancer, they might exist, but not in, solid organ fibrosis or in the bone marrow. It's basically resident mesenchymal cells that produce all the scar. And we are trying to understand what are the crosstalk partners, so which cells activate them to produce all this blue stuff here, the scar. And it's really similar to what you have when you cut your skin, right? You get fibrosis, you get scar formation. It's needed in your skin. It's needed when you had surgery. But the idea in these organs is that, chronic injury leads to this scar formation, and then this scar formation becomes a disease by itself that slowly destroys the architecture of the organ and then drives organ failure. So when we think really, we have really strong mouse data in this direction, when we can inhibit these cells from producing this scar, that we can actually slow down, progression of organ failure. So like chronic kidney disease patients, we can slow down their kidney functional decline. Heart failure patients, we can slow down the functional decline, that the heart really, stays pumping. And in the bone marrow, the same, we can slow down organ failure of the bone marrow, that the bone marrow still produces blood.
When you say resident, you mean native, we're born with
Yeah, resident means like
fibroblasts.
not circulating. There has been like some controversy whether there's certain circulating cells, like here bone marrow derived cells and fibrocytes, these are hematopoietic cells, and people have said, okay, maybe they actually, after injury to organ, they come from the blood, migrate in the organ, but it has been shown that this is not the case. These are resident cells, so they are localized in the organ, they get injured, and then they respond.
Yes, sir.
Do the signals that either of you detect, either in the marrow or muscle or kidney, are they detectable in the peripheral blood in an analogous way, so that you really can use the blood essentially as a biomarker for the kinds of work you're doing?
Yeah, that's an excellent question. And in particular for the bone marrow, this is exactly what we do and what we see, that you see the changes actually also in the blood, not only in the bone marrow, because they circulate, and in particular the endothelium, so the vessels, they get more leaky at some point, in particular in the disease, and then actually more gets into the bloodstream. And then we see the changes in the blood that we also see in the bone marrow.
So there's no work to the point that it's conceivable to use a blood analysis as a biomarker for the inception of disease or its progress.
Yeah, it looks quite promising. So you have to go to quite big cohorts to robustly test this, and that it's really something that's specific to the disease and doesn't have anything to do with comorbidities or other diseases or with age and so on. So we have to look at really huge patient data that we have to collect also, but it looks quite promising. So I will also show this. We have one marker where we think we can use this really as a progression marker.
Yeah, for kidney and heart, it's we're not there yet. So it's like, I mean, as a nephrologist, so I treat patients with kidney disease. We always think, okay, we have the urine, you know, other organs don't have the urine, so we should identify certain biomarkers that we can detect in the urine and say, okay, this is a high risk, you know, certain kidney disease patient, but this is something we can't yet. So we're trying to work on that. But so we are primary currently, and when you go to your doctor, the doctor realizes you have a kidney disease. Still, the gold standard is to take a little biopsy, a little piece of your kidney out, look at it under the microscope. So we are trying now to use some of these novel technologies to analyze the tissue and then subclassify the patient, risk stratify the patient, say, okay, this is a high risk patient, we have to change the therapy, things like that. So, but we're also looking obviously for biomarkers, but it's actually a lot of people are, but it's not very easy.
Fibrosis in the work that you're involved with. I know in the liver, certain therapies of hepatitis B and hepatitis C, there's some evidence that fibrosis that hasn't gone to an end stage will be able to be reversed. And corollary to that is do your technologies, these high throughput technologies that you do so beautifully, will they help in that analysis?
Yeah. Yeah.
Okay, maybe I can start. So like, we are currently focusing mostly on stopping or slowing down progression. So for the kidney and for the heart, there's not much data out there showing that it's actually reversible. There's some data when you have a diabetes patient and do a pancreas transplant, so you actually normalize blood sugar for long term, that there's some reversibility of the kidney fibrosis, but this is just like shown in a few patients. We're actually trying to do this in mice, so you can actually clamp the ureter, so basically the tube where the urine is transported from the kidney to the bladder, and then you get kidney fibrosis. So we're trying to unclamp to have some, reversibility, and then actually have a very, decent time course data set using these technologies to understand how much is reversible. And I think this is also something we wanted to come anyway to. That's why we actually look at these regenerative species here at MDI-BL, because some of them actually really are tremendously regenerative, and we want to understand why are they so regenerative, and what can we learn from human or mouse regeneration from these.
Yeah, and for the bone marrow, this is really a prime example of reversibility, because what you can do is allogenic stem cell transplant. So basically all the blood forming cells from the patient are removed, new stem cells are transplanted. And with this, you get really a reversal of fibrosis. So that's why kind of the concept was always, okay, you have to get rid of the diseased cells, get new cells. And so far, all the therapies have focused on in this here, you have actually a blood cancer that causes the fibrosis. And so all therapeutic concepts have focused on eliminating only the cancer, but not really on fibrosis driving cells. And so far, in terms of other therapies by drugs, this wasn't successful. And this is why we now really focus on the fibrosis driving cells, so the mesenchyme, basically, not only the blood forming cells. But in general, the bone marrow is a beautiful example that you can reverse even like this type of fibrosis.
Wait, I think over on the left side of the room, we had another question.
The trigger that makes the myofibroblasts is outside the bone or inside?
It's also inside the bone, and I think we have this on maybe the next slide. Yeah, this is what happens. So basically, we're looking at a blood cancer, and this is not an acute leukemia. So in acute leukemia, none of your the leukemic cells can differentiate. So it's just full of cells, but they can't make blood cells anymore. In this particular blood cancer, your bone marrow has just too many cells. And they produce blood cells, but you have just too many. So you have too many neutrophils, too many platelets. It's just that the body is too full of blood cells. And this can be controlled quite well. But the problem really is the moment these patients develop a fibrosis, and this is something you can see on the left side, the survival drops. So with just having the blood cancer, it's something that you can treat quite well. You can keep the cells in check. You can help them to differentiate, for example. But the moment fibrosis starts, this is getting a very aggressive disease. And then the survival drops from around 20 years to 1.5 to 2 years, which is more like an aggressive cancer. And the problem then is that your bone marrow is full of scar. There's no normal blood formation anymore. So at some point, really, the spleen and the liver start making blood, so they get really huge. And this is also, I mean, the patients have a really reduced quality of life, because you have not enough blood cells anymore. The spleen is big, so it's like also kind of skin itching is a problem. But then, of course, at some point, your spleen is so big it can't compensate anymore. And this is, so far, as I said, the only therapeutic option is an allogenic stem cell transplant. But most patients with this disease are not even eligible anymore because they're too sick, because it's quite an aggressive procedure. And there's no therapy so far that really has an anti-fibrotic effect. And this is like where our research started. We want to find something that really keeps the fibrosis driving cells in check, at least kind of stops the progression and better really leads to a reversal of fibrosis. And then at the same time, we also aim to identify biomarkers. And that was also already one question, to see if a patient is at risk, for example, to develop fibrosis. And also if there's like already a progression. At the moment, you always have to take a bone marrow biopsy. And this is quite an invasive and quite a painful diagnostic option. And it would be much easier to, patients come to the doctor every 6 months and then just take blood, look for a marker and see if there's like a rise, and then identify that there's a higher risk, and then maybe on top take a biopsy, but to have some feeling, because our understanding is the earlier you can, the earlier you diagnose fibrosis, the higher the chances that you can actually reduce it or stop it from progressing.
Okay, maybe we go to the next slide. So this is like, now we had this explanation of bone marrow fibrosis. So in kidney fibrosis, it's really that we think kidney fibrosis is what drives chronic kidney disease, which is really like a pandemic. So, when you look at the estimated global cause of mortality, chronic kidney disease is estimated to be the top 5 killer in the world by 2040. And 1 in 3 patients in Europe, or 1 in 3 people in Europe are at risk. And it's very similar in the US. It's currently the prevalence, so the amount of, people that are affected by kidney disease is already 10% globally. And so there's now some new therapies. You probably have heard from some, so you can do antihypertensive therapies. There's now these new drugs that are called SLG2 inhibitors that are mostly developed for diabetes. They actually slow down progression, but still the majority of patients, when they are diagnosed, progress with the disease. And this is really where we think antifibrotic therapies could help. So that's why we actually do this work, to identify what drives scar formation in the kidneys. Can we intervene there? Okay. So now this is 1 to 4, I think. There. So this is really, I think you're probably going to start with the bone marrow here, the mechanism.
Basically what we want to show in this slide is that there are a lot of conserved mechanisms, in particular in the cells that drive fibrosis, but the injury is different. So if you look into the bone marrow, the fibrosis, the injury that happens there is the blood cancer. So the blood forming stem cells that make all of our blood cells, they acquire a mutation. These are quite specific in this disease, and then they transform and and somehow activate cells that are not blood forming, that are sitting there in the bone. And our idea was to identify these cells and use them as, potentially as a therapeutic target later, and also understand how these two cells talk to each other. Because you can imagine, in particular in this therapy that you can use with a stem cell transplantation, if you remove the stem cells and then the fibrosis gets better, we think that you have to understand how they talk to each other and what you actually take away when you kind of take away the old blood forming cells and get new ones. And that was the idea.
And in the kidney, it's very similar. So in the bone marrow, you have these two compartments, basically here, hematopoietic cells and mesenchymal cells. It's very similar in the kidney. You have hematopoietic cells, or the immune cells, and you have mesenchymal cells. So these are basically the mesenchymal cells of fibroblast parasite in the kidney. The kidney also has these epithelial cells. They basically transport your urine, so after filtration. And this is actually what we think is happening in the kidney. You have injury of these epithelial cells, for instance, from protein leaking through the glomerular filter. You have protein in your urine. You usually just detect this first when you go to the toilet and there's some foam that builds in the toilet. So you have, this is an early sign for protein in the urine, which shouldn't be there. And then this injures actually the epithelial cells. They flatten, they change their behavior, they activate immune cells, they activate these resident cells. And this is really something we are trying to dissect using these technologies to say, okay, what are the ligands that are secreted from these epithelial cells that activate immune cells? What are the ligands that are activating mesenchymal cells, and what genes really drive them, their differentiation process that the parasites or the fibroblasts say we're going to become these scar-forming myofibroblasts.
Factors in bone hypertrophic cells, and as knowing the factors that cause hypertrophy.
Yes, yeah, so exactly. So we want to identify really the factors that drive this, and what I will also show later is that some of them, resemble to kind of injury of the bone. So kind of like an actual fracture of the bone. So, there's a lot that's kind of more, let's say, processes that are also activated in regeneration, for example, that we can see.
So that's like usually first like collagen formation matrix, and then you get actually later even bone formation in the disease.
Yeah.
I think that's one of the answers.
I agree, yeah, that's one part of it. In the heart, we have also hypertrophic responses. The cardiomyocytes get bigger. So that's also part of this picture that we try to understand what signals are actually activating the cardiomyocytes to grow.
If you both work in different organs and yet you're focusing on the same process, is it fair to assume that there are similar molecules, cytokines, or what have you, whether it's kidney, heart, or marrow, that is promoting fibrosis?
Absolutely. We see an incredible overlap. And in particular, we thought when we started this research that in particular for the bone marrow, it would be different because there you, for example, don't have an epithelial layer. So it's quite, it's organized in a very different way. But the cells that actually make fibrosis in the end, they are quite conserved and they look very similar when you look across different organs, because probably like even in the embryonic development, they're kind of like they come from the same place.
Yeah, exactly. So basically, there's epithelial organs like the lung, like the liver, like the kidney. So there, this triangle basically stands. So these injured epithelial cells in the lung, the liver, the kidney drive disease. And then you have these two major players, and these are very similar: immune cells, hematopoietic cells, mesenchymal cells. There's like, in the heart, you could just replace these epithelial cells here with cardiomyocytes, and you would end up with the same triangle. And then there's differences, obviously. These driver mutations that drive malignant hematopoietic cells in the bone marrow, this is not existing. But we also interestingly find that certain, these patients actually with malignant transformation of hematopoietic cells, they also get suddenly then some issues in the kidney and the heart. So there's also even crosstalk between the organs. But for instance, when you have chronic kidney disease, you also get
fibrosis in the heart. So there's really crosstalk between the organs, and there's a lot of similarities, particularly what we always see, very similar ligands between the immune cells and the fibroblasts that actually then become myofibroblasts.
So you've identified many of the sort of basic elements that there's a lot of cross insights from one organ system to another. Show us what happens when you orchestrate all of these tools at once to look at these systems.
Okay, I think we have here some data on the bone marrow.
Yeah, we can almost skip the slide. It just shows the diversity of non-blood forming cells in the bone marrow. And this was the starting point to use these technologies where you look really at single cells, because in the bone marrow, if you asked the question around kind of an audience, even in hematology, what are the cells that drive fibrosis? Everyone would give you a different answer or would give you a different name, maybe meaning the same cell. And then we thought this is exactly the technology we need here. We want to kind of understand what's the composition. So get kind of like insights in all these different cells. And then basically ask the question, are they all the same? Is it one cell type or are there hundreds? And so really provide names to even small populations. And this is exactly where these single cell technologies helped. And in the next couple of slides, we will show how this helped to even identify mechanisms that are important for the disease. Yeah, and what we can use, and we talked about, sorry.
So can you target the fibrotic tissue itself? In other words, the fibrosis itself is made up of the material.
The material. Yeah, we are looking into that at the moment, and it's also not super easy, but exactly to look also how the matrix changes, how the stiffness of a tissue changes, and in particular in the bone marrow, it's kind of let's say more, you would imagine it to be more free floating. And what happens at the point when you have fibrosis, the cells get stuck there, and you can even see that some cells that are still healthy, they die within the matrix, and that's kind of the same also in the heart. When you have areas where there's like a lot of matrix building up, also the blood supply is not sufficient anymore. So like loosening up, and we see factors, for example, these matrix metalloproteinases that eat, let's say, matrix that might also be attractive targets.
So I want to kind of try to move along a little bit because we are running a little bit out of time. So maybe if we could move through the slides, and then I want to ask you some questions just about your experience here. And then hopefully we can be probably in a little bit to talk about some of the conversations that you all have been having since you've been here.
That's good, yeah.
Yeah, so this is just showing there are different models. You can use, we mostly use mouse models so far that can really recapitulate the disease. So you see these massive fibers in a mouse, and we also get patient samples at different grades of fibrosis, so not so severe versus very severe, and even some after therapy to kind of compare. That's what we use the single cell approaches on. And then when we do this approach of looking at single cells, we could really identify the cells in the bone marrow that are not blood forming cells. And we identified a small population, so they're called mesenchymal stroma cells here, and they can give rise also to fat cells and to bone cells, so they have quite some plasticity. And we identified these small subset here, MSC1 and 2, as the fibrosis driving cells. So only these cells, when you have fibrosis, and that was true in the mouse, but also in patients, they start making matrix, and thus for us were the most attractive target.
Yeah, and in the kidney, it's very similar. So this is actually a diffusion map. It's a computational model. I mentioned this earlier. So every dot here represents one cell. And this is, I think, from around 10 patients in total. And using these models and all these different cells, so we detect around 3,000-4,000 genes in every single dot here. And then we can actually really ask what for differentiation processes are happening. So we actually discovered these are the cells here, the myofibroblasts that drive disease. And then we could really model that they are most likely derived from 3 distinct populations, 1 parasite population, 2 distinct fibroblast populations. And then when you actually look at collagen, which is a very important matrix protein that is really forming this fibrosis, you can see that these pericytes, fibroblasts and fibroblasts, actually, you see they are more white or greenish here. They don't express much of the collagen, but then when they differentiate towards these myofibroblasts, they get red and they acquire collagen expression. And this is the same for when you look at all ECM. So now you can also look at all ECM genes. So all collagens, all proteoglycans, all glycoproteins. You can see they all increase alongside this differentiation process. And then you can really do a lot of things with this data. We can actually add spatial information. So this is basically a scar in the human heart. This is U-shaped. You can see that here. You can actually now also map gene expression in space. So you can actually take the tissue, you actually take the mRNA out of the tissue, you barcode every spot in the tissue that you can actually computationally build back the tissue, and then you can really look at signaling pathways. Here's TGF-beta signaling now in space. It's really increased within the scar. You can look at the entire pathway. You can look at transcription factors that actually drive TGF-beta signaling downstream. You can look at their binding activity to the DNA using footprinting, where you know, okay, this is the binding motif of the transcription factor at the DNA, so it's more active or not. You can really map this back to space. You can see this U-shaped scar has increased activity of this transcription factor, and then you can look at even downstream genes that are regulated from this transcription factor. And doing this is just an example for this one pathway. You can do this with all pathways, and this really helps you to understand what is happening where in the tissue.
And what does that get you? So now, before you knew that it was happening, now you can see where and when it's happening at all these, in all of these different kinds of views, whether it's proteins or genes or other activities. What does that give you?
Yeah, so you're adding the spatial component, basically can identify certain cell populations that are in specific disease driving areas. So which cells are close to the scar, which are further away, which immune cells are close. And this helps you to really also look at crosstalk mechanisms. If there's spatially one specific subset of immune cells always co-localizing in space with certain fibroblasts, you can actually look at the crosstalk mechanisms and ask, okay, which ligands are expressed on cell type A, which downstream mechanisms in B. Is this something where we can actually build an antibody to stop this?
So proximity in space and time matters.
Yeah, exactly.
Thank you.
Okay, and then we actually, I think I mentioned that, then we really move towards validation. And, we can use organoids, for instance, so we can actually have this human-induced pluripotent stem cells that you can differentiate towards these mini kidneys in the dish. And this is also something that is done here. And then you can actually have, this really looks like a human kidney a little bit, but this is really just stem cells in the dish. So it's a human mini kidney in the dish. You can induce injury. You can actually add chips, and then you can add perfusion. You get actually endothelial cells growing in there. You can actually induce injury, get fibrosis, and then actually intervene. So this is something we try to use to validate some of the mechanisms in addition to animal models.
Yeah,
Okay.
and then we are going back to the kidney. So in the slide through the bone marrow, sorry. And so in the slides before, I showed you that we identified these specific small subpopulation of cells to be the cells that actually make fibrosis and make the matrix. And then we were wondering, are there mechanisms or genes that are upregulated specifically in the disease? So something that's not happening under normal conditions, but only if you have fibrosis. And then we identified a gene complex, it's S108 and S109. They are also called alarmins to be upregulated specifically in these cells, and they're not expressed at all under normal conditions. And then we also started validating, and specifically also if that's something we could see in the blood, because these are kind of, they're called danger-associated molecular patterns. So we thought this is kind of like an alarm in the end in the bone marrow, and we wanted to see if this is reflected in the blood. And there we could see that in the blood of patients with this disease, there was actually a correlation to the fibrosis grade. So the higher the S108 grade was in the bone marrow, but also in the blood, this correlated to fibrosis. So there we think this is really attractive to be a biomarker for the disease, and in particular for the disease progression. But then specifically, we wanted to inhibit this. And I said before, so we can use genetic knockouts, for example, so we can downregulate a gene or upregulate a gene. And that's what we have done. But we also checked the literature if there was already a drug out there that was targeting this process. And we found a drug, it's called tasquinimod, and it was tested in prostate cancer, and it blocks exactly these alarmins binding to a receptor called TLR4. So we thought, okay, let's repurpose this drug and use it for this particular model, in particular as it was tested in patients and it was tolerated quite well. It was never approved. It never made it to the market because it did not improve the survival in prostate cancer. But still, there was at the time like a reduction in the tumor growth and so on. So even there, it looked promising at some points. And now actually too far. So that's kind of like we tested it first, of course, in a mouse model. And as I said before, kind of in the disease, you get huge spleens. That's also what the mice have. So this is the disease model. The spleens are huge and they have like a large grade. So more than 80% of the bone marrow were fibrotic. And then when you treat it with this drug called tasquinimod, you can see the spleens look exactly like the control. So there was definitely an amelioration there. And we also significantly reduced fibrosis. And these data was so exciting that we started talking to clinicians in Germany, but also in the Netherlands. And this was something that we could translate into a clinical trial. So we are writing the study protocol at the moment. It will be a whole one trial, which is a trial in the Netherlands, Belgium, and also in Germany. And we will focus on patients that are left so far without therapeutic options. And we really hope that we will see the same effects that we have seen in our mouse models also in these.
So you took the information that you found from about the potential target that you did using a multiomic approach, and then you used that finding and interrogated data sets on what molecules might already be out there. And that's how you found tasquinimod, is it called?
No, the tasquinimod was really kind of, I mean, you can call it almost a Google search, just like looking up. So you look alarmins, inhibitors, I mean, really kind of just checking if there's like something out there and published. And then we found kind of this inhibitor that was focused on these alarmins. And there were other trials kind of like going a little bit more in rheumatoid arthritis, so something that also has to do with like inflammation. And yeah, that's how we found this drug.
Okay, so I'd like to pause for a minute, and because we only have 9 minutes officially left, although we might be able to come back to the slides in a little bit. And there's one particular set that really, for the layman, really sort of blooms the sense of what bringing all these two tools to bear, the visual sense. But I also wanted to ask you, why are you here at the lab? You're visiting scientist for your second time. I imagine Dr. Haller is a big part of your first visit, but something about it brought you back. What is this experience like for you? Both as scientists and just as people in the world.
No, I think it's really like, I mean, I'm a nephrologist, and this like MDI, I think it's really like has a, very strong nephrology or kidney disease history. And also, I think the species that actually are used here, we don't have access to everywhere else, particularly not where we are. So like looking at, zebrafish and looking at axolotl and stuff like that, where you have really regeneration, where you don't have scar formation. It's really some interesting comparative biology you can do to ask, okay, you have scar formation in mammals, in mouse and human, and you don't have this in these other species. So what's different in these species? Can this, help us to understand what we can do better in human tissue regeneration?
Well, please.
Yeah, no, I just wanted to add, for me, it's really, I think it's important to think also outside the box, not only to stay within your field, but to kind of talk to as many people as possible. In particular, we have also seen processes that are more important, for example, in regeneration. So, I mean, just hearing about models of regenerative animal models and so on just helps the understanding. And sometimes there are things that you don't see, or you think it's like maybe not so important. And
maybe we can bring Dr. Mirawala up. Prag Mirawala, who works in the axolotl salamanders. And I think Dr. Mirawala, you're also doing some multi-omic works. what's the conversation between you folks like? What are you talking about just this past week?
Yeah, so the microphone is working, that's great. So what we study is one more organ, and that is skin. In axolotl, we are interested in skin biology. Like when you do the injury in axolotl, they heal their wounds perfectly. They never produce fibrotic scars. So we are looking the same problem, but from a different angle. And that gives us a very unique perspective. And on a longer run, we also want to collaborate on kidney and bone, because axolotls not only regenerate their wounds scarlessly, but they also can regenerate their limbs. When you're regenerating a limb, you have to form the bone, you have to form cells that reside in the bone. Similarly, we are interested in kidney regeneration because they can also do kidney regeneration. So without undergoing fibrosis, what is so special in axolotl that allows them to regenerate all these body parts? That is the key of what we are studying. We also use similar approaches as Rebekka and Rafael, basically. We also use all the single cell transcriptomics and other omics approaches to understand what are the cells, which cells are the key to the regeneration, and how do they get activated. While in the case of mammals, we understand that hematopoietic cells can influence fibroblast for the fibrosis. In case of axolotl, the same hematopoietic lineage actually tells fibroblast to do the regeneration. So while the communication between two populations is same, why the outcome is different? And these are the key, and these are the kind of questions and topics that we discuss when we sit together over a beer.
Are you thinking of new ways to address each other's problems because of these crosstalks, these cross currents?
Yeah, exactly. So I think we're actually trying to design certain experiments where we really can compare what's different between these organisms and what can we learn from each other to understand biology, to at the end really to also learn from these species for human disease and potential therapies.
And so you see, so this is right on the spine of the way this laboratory is operated as a comparative biology center for more than a century now. Do you see, and a lot of this has been basic research, but it sounds like you see real translational pathways. And Dr. Mirala, you're doing some really basic research, but do you get excited also about the translational possibilities and the power?
Absolutely. Absolutely. I mean, see, one of the things that we are discussing at the moment, for example, if we take kidney and let's say you do the same kind of injury in axolotl and in mouse. And in one case, you have a perfect tissue regeneration. So kidney will regenerate. They will reform your tubules. They will reform all other organs or the tissue types or cell types that is existing in mammalian kidney. So how do these cells upon injury start talking with each other so that they can do tissue regeneration? And you have the same cell type in mouse, and there we try to understand, instead of doing the tissue regeneration, why are they moving toward fibrosis? So putting them side by side and comparing them is definitely going to help us understand some of the differences that underlie these different outcomes that happens between two model organisms.
So actually, we might be able to leverage one day, or maybe in a year, know a little bit more about how to leverage what it is about axolotls that allows them to
regenerate,
to regenerate without scarring, or vice versa, and maybe get towards a new human therapy, or maybe, I mean, do axolotls need therapy drugs to help them?
Yeah, so
look at it the other way.
Well, I'm sure people are not interested to treat axolotl, and also axolotls are a fixed problem. They don't produce scar. They don't lose. I mean, they can lose organ, but they will regenerate those organs. So there is nothing to fix there. They are marvelous. But what we want to achieve is we want to learn some of these principles. So when we think about certain pathways, for example, they mentioned TGF-beta pathway. TGF-beta pathway in case of mammals leads to fibrosis. But when it comes to axolotl, that same pathway is essential for tissue regeneration. So this tells us that, okay, the same pathway you are deploying in both the species, but the outcomes are different, which means that there is something else that we are missing to understand why things are going wrong when we have mammalian cells.
I think we've come to a good moment now. And Dr. Haller, you know, this seems to get to the point of why do we want, what do you need from our visiting scientists?
Well, first of all, it's great that they are here. But then I think we all realize that there are two major changes when you compare this to before. Rafael mentioned that we have a long history of kidney, a long history of transport and all that. But for the first time, really, we talk about biomedical mechanisms. So we're talking about fibrosis. This has not been a topic at the lab for a long time. We were interested in transport, but now we want to understand in the one animals where it doesn't seem to be a problem. Do they have any problems at all? I mean, axolotl should be the happiest animal on the planet. I mean, no tablets. No, but that's one major thing. And the other is really what I mentioned before, that I'm used to look through a microscope, but these guys are looking on a subcellular level at mechanisms and playing around with lots of molecules. The numbers are staggering, and this is also something very new. It's not only happening at MDI-BL, but this is also a change. And besides having the experience of the Kramann family and their wonderful children, and it's great to have them here to start all these discussions.
Thank you so much for coming. Rafael Kramann, Rebekka Schneider. Jerry?
I'm sorry, I was just going to ask you guys if you could tell us a little bit about where you see the future of this going, right? So Dr. Haller mentioned this is relatively new, this idea of using big data. What does it look like 10 years from now? I'm just, I mean, I know that's impossible to really articulate, but I'm just curious.
I mean, my vision actually for the kidney field is like in like 10 years from now, maybe using all these data sets, you can actually do a dipstick from the urine and say, okay, this is a high risk IgA nephropathy, so like a certain kidney disease, and you need to either have a new therapy or you can allocate this patient to immunosuppression that is available.
Okay, but now to get a little bit further, because every scientist thinks that they solve the world, and at the moment we have all this. So my challenge would be, you have all this told us about genes and especially proteins and all that. So we are a little bit interested in sugars, and we have just talked to Ron Costanche from the chat, and he's telling me that lipids is everything which is important. So how are we going to deal with sugars and lipids in your protein-orientated world in the future?
No, I mean, I think the field is, you know, these omic technologies and things, particularly looking at single cells, is tremendously rapidly really changing. There's like new tools developed every week, basically. So I think we're just at the beginning of all of this, but it still helps us to look at tissue injury, repair, fibrosis, with, you know, having much more information out of tissue and data sets. And I think this will change more. So I think there will be much better understanding. But when you look like, you know, research like 50 years ago or 30 years ago, which was more hypothesis driven, this is really different. We do now this really different in our lab. So we start with this data set, and when we identify novel things, and then we try to validate them. So we don't have an hypothesis initially. We just start with the big data and then build hypotheses with the data. And I think this is really changing how we approach science.
We have so someone had their hand up earlier. I just want to ask you guys, you have 4.5-year-old twins here on campus. Is your babysitter still with us?
Yeah. They're great. Yeah. Thank you.
I do.
You must have a dream bucket. If you had a few $1 million, what instrument or what tools do you need here to move this fast? Do you need people? Do you need organizational change? Do you need money? Do you have a vision of the tools that are available worldwide to tap into all the intellectual work that's being done around the world in this area, so that the brain center can be here and communicated with thousands of researchers that are working on kidneys, for example.
No, very good question. So like certainly I think people are needed, to have some, and I think the campus is like really transforming tremendously, having round the year research people here, so that people, and then I think there's like really tremendous work on new technology. So there's a lot of very good equipment here, imaging wise for microscopes and also single cell genomics. I think there's some other layers. Yeah, exactly.
I'm talking about molecular.
Yeah, like the spatial technologies, there's like really like a development towards spatial, where you have novel tools and instruments. That would be really fascinating to have.
Devices.
Exactly, yeah. Like this is currently still sent off, I think, for sequencing. This is certainly something you can also do, in-house and then speed up things.
Yeah.
Right.
You want to go ahead?
Along the same line, because we were asking about it. So how can we link with the greater community out there? Is there a level where you can talk about this? How do you validate your systems with other people? How do you exchange this? Is this only via computer?
Yeah, I mean, open data, open science, I think it's really, really helping the human cell at last, and this can be done for other organisms as well. You regenerate these single cell spatial tissue maps in homeostasis and disease, and you make this data accessible to everyone in the world. And then you have really communities like the human cell at last now, where you meet and discuss this and, share the data that, everybody can access the axolotl single cell data in injury and compare this to, whatever human disease or mouse injury model you have to really, have this comparative biology available and learn from it to how to basically, eventually develop therapies.
So I'm just going to ask you, maybe if you could show us those last few slides.
Okay.
Okay, great. So I think, Anna, is there anyone online who's got a question? Okay, so I think we've had a pretty informative, and I personally will have to go back and view it again to make sure I understand absolutely everything that was said, and I hope that others will do the same. Thanks very much, everyone.
Thanks so much. Thanks to all of you for a great presentation. It was very exciting and fun to think about what the future looks like. I think it's an amazing project that you've taken on. Not simple for sure, really complex, but wonderful. Just a quick note to let all of you know that, let's see, our next Science Cafe will be on July 24th, which is actually a special lecture with Dr. David Evans. That entire week is a fun, what we're calling homecoming week. It's really just an opportunity to celebrate the 125th anniversary of MDI-BL. So we have lots of great activities planned. Dr. Evans will be focusing on just, 125 years of discovery that's been happening here at the MDI Biolab. So we encourage you to join us for that. In the meantime, this coming Wednesday, we have Family Science Night here on our campus. So it's a wonderful opportunity. We tend to focus on younger ages, but everyone is more than welcome, and it's a lot of fun. You'll have an opportunity to talk with our students and learn a bit about some of the work that's happening here in a fun way. Plus, we're going to give cookies that have the lab logo, so you don't want to miss those. And there's an ice cream truck, which is my favorite. But anyway, we really look forward to having you join us. If you're super bored, I'm going to be at the Bath Harbor Library on Thursday talking a bit about the lab history. So if you want to trek out that way, we'd love to have you join us there. But thank you all for being here, and thank you for being such great supporters of Science Cafes. And thank you so much to Dr. Kramann and Dr. Schneider for being here with us, and Dr. Merwala. Thank you. All right. Take care, everyone. Thanks a lot. Yeah.